Thank you. Well, it's interesting you say reality-based, because one of my appointments is actually still at the White House. That's just this reality-based institution these days. So actually, with that in mind, I have to introduce some very important disclaimers. So I do retain an appointment at the White House. However, this is not to be construed as an official White House talk by any means whatsoever. I'm required to say this every time I give this talk because I guarantee you this was not approved by White House communications, and that this is a personal talk. This talk may or may not represent the views of the president or the administration. What I can tell you, though, is that they certainly used to. Yeah, if you want to know the official statements or official policy of the administration these days, I'm told literally that they're available on Twitter. So yeah, it's amazing how fast that slide became out of date. All right, the other disclaimer is that I'm not an expert in machine learning. I am a computer scientist, or at least I used to be till like 6 weeks ago. But even there, I did programming languages, and I did programming languages applied to stuff like cybersecurity and domain-specific languages for supercomputing and things like that. So I'm not even your kind of computer scientist either. Yeah, so what I'd like to do now is define precision medicine. I'm about to define an algorithm for precision medicine, so we probably define precision medicine first. And I think my old boss really got it right when he launched the Precision Medicine Initiative with a definition for what it is we're trying to do here. And so for him, precision medicine was all about doing data-driven medicine. And in this case, it's often guided by the genome. People are very excited about all the data available in the human genome, and justifiably so. There's a lot you can do with that. But it's not just the genome. There's all forms of data that matter here. And it's really, at the end of the day, about delivering the right drug to the right patient at the right time. That is the essence of precision medicine. So with that in mind, I also want to highlight that I think one key component of precision medicine is that when you do precision medicine in the limit, as I'm going to talk about today, you end up transforming science into medicine. Because sometimes that's really all you can do for a patient is actually advance the science in the direction of what it is that they need. And so there's three key things I want to get across in my talk today. I want to tell you a story. It's a personal story of what happens when you're told that your child's the first case ever discovered of an ultra-rare genetic disorder, and really the aftermath of that. And really what I'm going to do with that story is illustrate a concrete execution of a particular algorithm that I've been building for precision medicine. So really a high-level process for taking patients to the right treatment. The other thing I want to do is call out opportunities for machine learning that I see popping up in the context of precision medicine. I think there's quite a few. And actually, I'm glad you mentioned clinical trials because I think that's one of the biggest ones. I'll touch on clinical trials at the end. So let's talk about the algorithm. I mean, the idea here is we want to take a patient, we want to transform them into a bunch of data. We want to identify what's wrong with them, and then use this data to find the perfect treatment for them. This is the goal of precision medicine. It's ultra-individualized care. And so really we want to do this in a very robust, scalable way, which means we have to computationalize it. We have to come up with a function that maps patients to pills. But this is actually quite difficult to do. So really what we want to do is come up with an approximation of this process so that we either map them to a pill or to some sort of experiments you have to run that might give you insight onto what the best possible medication is. That's really the high-level idea behind this algorithm. So let's start with my input to the algorithm, which is actually my son, Bertrand. Yeah, so he's now 9 years old. I think this is when he was like a year and a half. And like I mentioned, he was the very first case ever discovered this ultra-rare genetic disorder. So here's how the process unfolded for us and how it does for so many other families. And I know this is a story about my son, but this is by no means unique. There's a ton of people going through this exact process. So with that in mind, it begins for almost everybody at a place called Undiagnosed Island. It's an island with a population of one patient or one family. And for us, it was a tough place to be because we didn't know why while we were stuck here. My son was having seizures or developmental delay or movement disorder, or why for some strange reason he could not cry tears. And he could cry, but like no liquid would ever come out of his eyes. And we were stuck on this island for 4 years. For 4 years, we had no idea what was going on. I mean, it's not for lack of looking either. We took it for almost every specialist in the country. And ultimately, it was a team of scientists at Duke University that said, we are willing to run the experiment that we think will give your child an answer. So I've been learning enough about genetics to know that I'm pretty sure this is genetic, and I'm pretty sure to find it by this point, we need to do genomic sequencing of some kind to figure out what this is. And that's what we did. So we did trio exome sequencing where they looked at me, they looked at my wife, they looked at my son, they said, okay, well, what is this? And what they found at the end of this process were 2 loss-of-function mutations in a gene called NGLY1. So he had wiped out function in this gene. And then they said some very unusual stuff. They said, it looks like he's the first, and in fact, only patient we've ever seen that has this disease. In fact, we're not even sure if this is the cause. There's other mutations in here. This just looks really suspicious. And if this is it, he really is an N of 1. He's the first patient to ever have, at least recorded to have this disease. So the question becomes, well, how do you know that this really was it? I mean, there was dozens of mutations that looked really suspicious. Why did we focus on this one? Well, in this case, what we did is we had to take all of his phenotypic data in the form of his health records and map that back onto the underlying purported genotype and see if there's a fit. In this case, then we'd actually understand what this missing gene was doing. And in this case, NGLY1 is a gene, it encodes an enzyme called N-glycanase, and N-glycanase is responsible for deglycosylating misfolded glycoproteins after they've been retrotranslated from the endoplasmic reticulum into the cytoplasm. I guarantee you that as a parent, there are certain sentences you have to expect to say someday. That was not one of them. So anyway, it's a recycling enzyme. Our view is actually evolving quite a bit on what it really does, but it is a recycling enzyme for decommissioning these misfolded glycoproteins. If it goes away, then what you'd expect to see is this trash pile up. The trash in this case is misfolded glycoproteins. So we said, okay, well, if this is what it's supposed to be doing, can we find evidence of that? And we actually poked around in his medical records and found some evidence from a liver biopsy that looked like this really was happening. And said, okay, well, this is almost certainly what's going on with this disease. Almost certainly. So I think here's the first opportunity. And this is already to some extent being done, but I don't think it's being done particularly well yet, which is predicting the pathogenicity of individual mutations. So given a mutation, because this happens in cancer, it happens in rare disease, given a list of mutations, which of these are the ones that are medically relevant? Which are the ones that are actually driving disease? And so I think there's a huge opportunity for machine learning here to get this right. And right now I can tell you that when you use these algorithms that exist today clinically, they get it wrong a lot. And this matters for patient healthcare. And of course they say this is not actionable. There's nothing you can do when you're the first patient in the world. And we literally can't even give you a prognosis, which is frustrating. But the thing is, I just inherently didn't believe that. I don't think it's actually ever been true that something's truly not actionable, because we went from dozens of potential targets, all of which were wrong, down to just one target overnight. And if you give a parent who's inherently motivated a target to shoot at it, I guarantee you they will start collecting arrows. It's just what we do. So it was literally weeks after the diagnosis, we were actually looking for things to do. And I think the key theme here is that even though there was no medication to take, science itself becomes a kind of action that you can take. Science itself becomes medicine in the event of the not actionable. So the very first attack was launched, I think, literally just a month after getting the diagnosis. One of the mutations he got from me was what's called a premature stop, meaning there's just basically a stop sign in the middle of this gene, which prevents formation of the full protein. Well, there's medications out there which are supposed to be able to override the stop sign and give you the full construction of the protein. These are called read-through compounds. And we were testing them on his cells almost immediately. Now, unfortunately, none of them actually worked. This for me was a sobering experience coming from computer science, where generally if things work in theory in computer science, they tend to work in practice too. Not the case in biology. It's a very different field. But still, it was motivating to think that we could take a scientifically credible attack on this disease just a month after figuring out what it was. So I said, well, let's do some more science. And of course, if you want to do science, you have to stand on the shoulders of giants. So let's go find the giants. Let's see who out there is working on this. And that's when we realized basically nobody. There's like 5 papers in all of PubMed back then on this gene, which is effectively nothing. So well, if we're going to do this, we definitely need more giants. And so what I did is I went out to San Diego to team up with this guy, Dr. Hudson Freeze. He's a glycobiologist at Stanford Burnham, who was the closest thing we could find to an expert, because he at least understood the pathways involved. We said, tell us what you think is happening here and where we need to go. And so what he did is he really took us to the peak of our diagnostic journey. He showed us how far we had come and gave us finally a sort of a perspective on the landscape that we'd traversed, but also ultimately how far we would have to go if what we wanted to do was reach understanding, treatment, and cure, which was significantly further still. So as challenging as those first 4 years had been, we realized that it's going to be significantly more challenging still if what we want to do is actually get a treatment into Bertrand. As a parent, you don't really think, okay, well, this is impossible or it's difficult. You just sort of say, oh, well, I'm just going to take, the next step that I can reasonably see to take and keep doing that every day. And over a number of years, it actually starts to add up to real progress. But I realized, right off the bat, there's no way you can do something like this alone. This is not a one-family kind of operation. So we have to find other patients that have this disease. I mean, I know that Bertrand is the first discovered patient, but clearly there are others out there somewhere. And the reason I knew that is I could look in these genomic databases at the time. I could look at the distribution of these pathogenic alleles or likely pathogenic alleles and do some back-of-the-envelope calculations to say that even though this is a 1 in millions kind of disease, there's probably around 500 people somewhere in the world that have it. They just don't know what they have, or their parents don't know. And so I said, well, I think we can find them. And to find them, I wrote a blog post. And I really wanted this blog post to do two things. I wanted it to go viral, so to sort of get in front of as many eyeballs as possible. And I also wanted to do search engine optimization, so that if other patients were out there Googling their symptoms, or their parents were out there Googling their symptoms, this was going to shoot up to the top and find them for us, or they would find us. So this is the blog post that I wrote. And so the title and the picture of Liam Neeson, that's designed to make it go viral. And I probably could put like cat pictures up there too, and it was done just as well. The internet is kind of sadly predictable with these things. There's like 5,000 words beneath it to handle the SEO component. And the crazy thing is it actually worked. So it did go viral, and it did actually start ranking very highly in Google search results. So it was seen by like millions of people in a very short amount of time. And sure enough, was popping up for all the right search terms, like lack of tears or NGLI1 or something else that somebody else might be typing in if they were looking at a diagnostic report or wondering what it was that their kid had. And so literally within weeks, patients across the world in Turkey were identified. They identified us. And over the course of the past 5 years, patients have been popping up all over the world. And this I think was a key moment for us to go from an N of 0 to an N of 1 to suddenly a community. Because what that did is it really washed away this dark night of uncertainty and replaced it with the sunlight of science and community. And so I can tell you today that this N of 1 has grown to a community that's very united and a common goal of understanding, treating, and curing this disease. And in fact, actually I need to update this too, because we have 59 patients as of last week. 59 patients around the world that are working together. And now we can ask questions like, well, what can you do, with this much bigger N that you couldn't do with an N of 1? So that there's a lot you can do once you have a real community to study. For example, you can do a natural history study. You can start to really understand what's going on with the phenotype of this disease. And we got very lucky that at the time this disease was being discovered, the NIH was actually setting up a natural history study for disorders of glycosylation, of which we reasonably argued this was one. It's really a disorder of deglycosylation, but we tweaked it enough that they said, okay, we'll let you in this study. So we basically took over this protocol at NIH. By patient volume, I think we make up like 80% of it. It's because we took every living patient we could find and pushed them through it. And Bill Gall, who actually runs the protocol, was very kind to let us actually do that. To give you a sense of what it's like to go through this protocol and to get really deep phenotyping data out of a patient, what I have here is a picture for every prod procedure or appointment that occurs during the course of a week-long admission to NIH. This is an admission that every patient has agreed to go through 1 week every year indefinitely. So this is an intense commitment on the part of the patient community. But what you get when you push everybody through like that is lots of data. And in fact, after running the protocol for just a couple of years, we already have clinically actionable data. So we found biomarkers, very clear biomarkers, sifting through all this data from these patients, including things like a urine oligosaccharide. So there's a unique oligosaccharide that occurs in the urine of these patients, which is considered diagnostic. And that diagnostic test is already available in the urine screens from Mayo and Emory. It was used by patients or by doctors at Boston Children's to diagnose a new patient for the first time using a non-genetic diagnosis just a few months ago too. So by having all these patients get in here and participate, we found biomarkers that are driving the discovery of more patients and giving us potential surrogate endpoints for when we go through clinical trial approvals with the FDA. So that we've done a lot with the community, but I think the real question is how do you go from understanding this disease, of which we have a very good understanding now, to actually treating it? And there's a lot of routes here. And so this is where it becomes a little bit more algorithmic, where I'm going to illustrate some of the paths through this algorithms on the road from the patient's data to a treatment for them. So the key thing here is you want to be screening drugs. You want to be coming up with ways to say, would this drug in any way be reasonable or effective for this disease? How do you do that? How do you generate this list of compounds? And how do you actually test them to see if they're actually doing something? So at a high level, the way drug screening works is you sort of take the patient, you look at their data, you model, you build some kind of model. It could be a cellular model, an animal model. Sometimes you have cell-free models, but you want to build some kind of model out of the patient's disease so that you can actually get a measurement of whether or not it's in a sick state or a well state. So By phenotyping, you can recognize all these cells, perhaps a cell line you created from the patients, as diseased. And what you're trying to do is combine these with as many compounds as you possibly can in order to figure out if any of them reverse the phenotype and make it happy again. Because that could actually be a treatment for the disease. It is not always the case that if it works in cells, that it works in people, but it's a start. This is sort of a high-level way, or a high-level view of how the screening process is typically conducted. To drive this, you really need to have good assays that are measuring whether or not you're hitting the mechanism for the disease in some way. And we were very lucky that for NGLY1 deficiency that what sort of the perfect assay showed up around the same time the disease is being discovered. There's an assay that measures D-glycosylation by this enzyme. And so we can check to see if there's anything out there that might restore it. We've got other assays now that look at different aspects of the disease, but we can actually run these on high-throughput machines. And actually, literally just yesterday, got a grant from NIH to do this at NCATS. So we're going to be running several drug screens at NCATS using these assays that we've developed for this disease to see if there's anything out there that might ultimately restore the fundamental mechanism or stuff that's downstream. There's another opportunity here. One of the biggest challenges when you discover these new diseases, because I've gone on to work on quite a bit more now, is how do you measure disease in these model systems? How do you measure whether you're hitting the mechanism in some way? How do you develop an assay? And I think there's definitely an opportunity for computational assays. In fact, there's a firm out there right now called Recursion, which is definitely using tons of machine learning, computer vision, all this sort of stuff to do morphological or morphometric assays in a high throughput fashion. So I think there's tons more opportunities in this space, though, because you can use more than just morphometric assays. You could look at RNA-seq or all kinds of other modalities for generating computational phenotypes. I think there's a lot left to do in this space, and we're going to need this if we want to be able to cure diseases at scale. If we want to have a lot of disease come through, we've got to get to this key bottleneck phase as quickly as possible. We need to very rapidly identify a phenotype in some model for the disease. Speaking of models, it turns out it's key to develop as many as you can, because you never know where the phenotype is going to show up most robustly. So for NGLY1, we have mice, we have flies, we have zebrafish, we have worms, and we even have yeast. And the amazing thing is, again, around the same time this disease was being discovered, CRISPR was coming out. And so the initial estimates on how long it would take to do this stuff was like, well, it's going to take a few years maybe. And suddenly, and also hundreds of thousands of dollars, and it dropped to like tens of thousands of dollars in a few months overnight. I mean, CRISPR changed everything so fast, it was crazy. And that's what has made it very easy to build all these model systems for this disease. Enzyme replacement is a strategy. And I think, in this case, you know what's missing. NGLY1 is missing. So can we synthesize the enzyme and just put it back in the cells? Can we use the enzyme itself as a drug? Well, sort of. You can, I guess. In fact, if you Google NGLY1, you can buy synthetic human NGLY1 on the internet. And it's only $402 for 10 micrograms. And in fact, you get same-day shipping in Boston. But if you extrapolate this out to therapeutic quantities, it's like $2 million a week. And so actually, I asked my old boss if this was covered under his healthcare plan. He said, no, Matt, it is not covered. But maybe. Certainly not on the new one either. So yeah, it's a bit expensive. And even if it weren't expensive, it's impractical. Because if you just inject this enzyme directly into the patients or have them eat it, it basically gets digested out of the lysosome. So you have to modify the enzyme in some way to turn it into a drug. In this case, you want to do something like pegylation, where you attach these polyethylene glycol anchors to it to reduce things like renal clearance. And also, to make it cell penetrant, to get it into the cytoplasm, you need to tip it with something like TAT. These peptides that are harvested from HIV, which make it cell penetrant, but which can make other things cell penetrant too. So it's an engineering process. And actually, there's probably some opportunities here for machine learning as well, because where you place these anchors is an optimization problem. So I'm sure there's some learning you could do here as well that might tell you this is probably the right place or the wrong place to put these anchors. Repurposing is a huge aspect of precision medicine. And so a general way to go about this process is to say you've got a patient, but let's imagine them as a vector, where this vector is a sequence of measurements of how they deviate from the norm in a number of dimensions. A common way of doing this right now, to the extent this is common at all, is to use RNA sequencing data to measure these deviations. And then to say, well, can we compute a drug cocktail that's the inverse of these disturbances? or if in case of expression data, is there some cocktail of drugs that produces the opposite disturbance in gene expression? Because if we can find that, then maybe we combine the drug in the patient and we sort of net out the disease. That's the idea. So yeah, I think there's a lot of opportunities for repurposing. In fact, I would say, most of what's going to happen in the space of precision medicine over the next decade is going to be largely driven by repurposing from the existing approved arsenal. May not all be done with RNA-seq or with a process like this, but I think that's going to be one of the key themes. And of course, there's some math behind selecting the optimal drug. We'll skip over that. Something else you can do is using the data available from the patient population itself to find potential treatments, because you always get a spectrum of phenotype. It's almost never the case that one disease presents uniformly, and that's certainly the case with NGY1 deficiency. So what you can do, even though these patient populations are small, is look at all the patients, look at all their genomes, and start to, if you rank them by, severity, you maybe start to pick out modifiers in the genome that are having an effect on the disease. And if you find these modifiers, they become drug targets, something that you can go after with drug screens. Something else we tried, and might actually be successful, is crowd screening. And by this, I mean, you just ask Twitter. Twitter, what would you do if you had this disease? And I tried this early on, and a postdoc working on cancer from Texas reached out and said, I've got a class of read-through compounds that aren't anywhere near market yet, or anywhere near patients at all, but I'd like to test them on your son's cells to see if they do anything. And sure enough, they do. They restore expression of the missing gene. This was 3 years ago. This is now, it's gone from a lab to an actual startup company to preclinical studies. It might actually hit the market at some point. So there's, this is a potential, actual read-through compound that if it does reach the market will likely work for my son. And then we've had, much more detailed efforts in this regard. So we've partnered with Andrew Sue's lab at Scripps, created a platform called Mark2Cure. What Mark2Cure does is it allows anybody with an interest to start annotating abstracts in such a way that you can build knowledge graphs and then mine those for potential relationships between treatment or between genes and symptoms and treatments. So when you're doing the Mark2Cure, you're being presented with abstracts like this, and you're just highlighting them in different ways. And you're highlighting genes, diseases, and treatments. And what you get at the end of this process is a graph like this. And this is a graph that's still going. The project's still active. We're still building this graph. And the very first graph that came back already had some very interesting paths in it, from NGY1 over to a compound here called ACTH. And the reason this was interesting to me, and what was interesting here is this was a path that goes from gene to phenotype back to treatment. And oddly enough, so even before Bertrand was diagnosed, he'd already been on this compound called ACTH, which was identified over here. And when you put these patients on ACTH, it actually completely wipes out their seizures. I mean, they go seizure-free on ACTH, from literally hundreds a day to nothing. And in many cases, they actually resume normal development. So this is Bertrand's EEG starting ACTH, and it almost completely normalized. I mean, I'm obviously not an epileptologist, but you can just look at the shape of the waveforms and see this looks much more normal. In fact, when neurologists would come by, they'd say this EEG looks completely normal. But that was found or sort of rediscovered in this knowledge graph, and there's other compounds we found in there since. And this is kind of amazing because when we put him on this, when he was about 2 years old, I would say, he actually started smiling for the first time. And imagine if we had found this, even earlier still. And I think there's more compounds in there even now to pull out, because we're still finding connections between NGY1 and other genes and other potential treatments by just mining this data. There's another opportunity here. And it's even being somewhat deployed today within Mark2Cure, which is to use machine learning and NLP to pre-annotate these abstracts. So instead of having to sort of annotate everything by hand, it actually takes a guess at what the annotations should be. So you're really just correcting the annotations, and of course that can go back and reinforce the next round of annotations as well. So there's something else you can consider in the context of precision medicine, which is metabolic diets. In many cases, what's knocked out or missing or malfunctioning is an enzyme. And enzymes are things that turn A's into B's. So if you suddenly are missing this thing that turns A's into B's, well, maybe the problem in the disease is you have too much A or you have not enough B. This is just sort of raw logic applied to the absence of an enzyme. And so there becomes a natural strategy to say, well, if you have too many A's or too few B's, maybe what you should do is you should supplement with B's. You should give B's, or you should find a way of restricting A's, the precursor to A. And there's examples of diseases where this works very well. So for example, for PKU, if you don't restrict phenylalanine, it's a devastating disease. If you do, you're effectively normal. Likewise, for MPI-CDG, if you provide mannose 6-phosphate, it's actually it's a very treatable disease. If you don't, it's a very devastating disease. So I mean, these are obviously very, very tailored dietary recommendations, where you know a specific genetic insult that leads you to an enzyme that says this is what you should probably do. Well, NGLY1 is an enzyme. It has A's and B's. Maybe we consider the same kind of thing here. So it's about 2 years after the discovery of the disease where I finally got into this kind of thinking and thought maybe we should try this. And by then we actually had enough understanding of the basic science to really understand what the A's and the B's ought to be. So putting everything together, I said, what are the A's and B's in NGLY1? Well, we know the A's are misfolded glycoproteins, but by the way that these things actually misfold and aggregate, they're sequestering a particular metabolite. There's a missing B, and the missing B in this case is an acetylglucosamine, and it's represented here by this little blue square. So of course I Googled it, found out that you can buy N-acetylglucosamine on Amazon, and I sort of reasoned that for what might be therapeutic quantities, it'd be about 25 cents a day. So I bought it. I took a bottle of it myself, and I didn't die. So I consider that to be effectively FDA safety testing, because in my household I am the FDA. So I have to remind you that I'm not actually the FDA. At the rate people are losing jobs these days, you never know. Then after a pretty serious hospitalization for Bertrand, I put him on. Yes, it was one of these ethical dilemmas. Like, okay, well, I'm pretty sure it's safe, but do I put Bertrand on this? And well, he answered that for us. He went to the hospital, came pretty close to dying, and I thought, okay, when he gets out of the hospital, I have to try this, because if he'd actually died in the hospital, I never tried this thing. It had been sitting on my shelf the whole time. There's no way I could forgive myself if I hadn't done that. So he gets out of the hospital, and after about 3 weeks, and he's sort of back to normal. I give it to him, and about 3 days after putting him on this compound, he cries his very first tear. So you know what you're going to see when you do something like this. But I saw that. I said, okay, well, something is happening. So I did what a parent would do when their child cries for the first time. I collected this tear, packed it on ice, and shipped it to a lab in California for analysis, where we did tear proteomics. So it was and of course he's cried many tears since, and other kids have gone on this compound. They cry tears too. So it started off as one small tear for Bertrand, but what it really did is it unlocked an ocean of science for the disorder. Because what I learned very quickly is that biologists are never content with it works. I always want to know why. They always say, well, it's great, Matt, that you found something that works in people. But what I want to know is, does this work in flies? And so I partnered with Clement Chow. We built a fly model of the disease. And what's fantastic is if you give flies NGLY1 deficiency, they die. They have about an 18% survival rate once you give them this disease. But if you put them on this compound that I found, they go up to about an 80-90% survival rate. And we've been able to use these flies to understand, really in detail what's happening with this particular supplement, because there's actually a lot that it's doing, more even beyond correcting this deficit, as it turns out. And the nice thing about flies is that you can actually cut them up and dissect them and analyze them, which you certainly can't do with the human patients. So they've yielded a lot of science about the disease. And this is where I started, shifting, because I said, well, okay, we found something, and it corrected part of the phenotype, but can we keep going? And this is where I think I personally started going more towards the biological side of the fence with some of the research I was doing. And actually picked up a small grant to do de novo drug development for NGLY1 deficiency. And at the time I was at the University of Utah, partnered with these folks here. And what we did is we built a planarian model of the disease. So planarian worms are awesome because if you cut them in half, what you get is 2 planaria. So they're a good model of developmental disorders because they have this regenerative phenotype. If you're looking at development, this is a pretty good phenotype for that. So we were hoping we'd lose it if we knocked out NGLY1. And the interesting thing is, if you give planarian worms NGLY1 deficiency and cut them in half, do you know what you get? You get a worm that's been cut in half. They have lost their ability to regenerate. So sure enough, we got the phenotype we're looking for. And then we looked for other genes we could knock out to see if they would actually restore the phenotype. Or restore this regenerative ability. And sure enough, if you knock out NGLY1 and another gene called NGase, then they gain the ability to regenerate once more. So we said, okay, well, what we have here now is a target to shoot at. What we want is an inhibitor for NGase. At least in the worms, it seems to be reversing this phenotype. And it turns out, if you do a double knockout in mice, where you knock out NGLY1 and NGAs in mice, they go from dying embryonically to surviving. So we've got multiple confirmations here that this is a strong target to go after for this disease. So that's what we did. This is where I started going back towards the computational again. So there wasn't a crystal structure available for NGAs at the time. So what we did is we used homology modeling to take the crystal structure for the bacterial version of NGAs, this target we wanted to shoot at, and tweaked it into what looked like, or what we guessed was the human form. So then we did docking simulations on this, where we just looked for molecules that are inverse in shape and charge to this catalytic domain here, so that they might bind it up and shut it down. That was the idea. And sure enough, after screening hundreds of thousands of molecules in these simulations, we found about 70 that looked like they might work, and 14 were FDA approved already. So then we took these 14 to the bench. We mass spec to see if any of them actually were true inhibitors, and sure enough, one of them really was. Really did hit the target, and that one, Prevacid. So you can literally buy Prevacid at Costco. It's an antacid, or it's an acid reflux medication. And we obviously don't care about the fact that it's an antacid or that's for acid reflux. It's all we really care about is the fact that it happens to have the right molecular shape to hit this other target, NGAs. And we actually found that all the proton pump inhibitors, of which this is a class, all have activity. So that molecular backbone that gives them this PPI activity also gives the NGAs inhibition. So published this a few months ago, actually, describing the process of identifying an NGAs inhibitor. And it's weird. I think of all the stuff I published in computer science, I'll never be as proud of any of that work as I am of this one paper right here, even though it's completely out of my original field of study. So there's another opportunity here, which is, I think, using machine learning to drive docking simulations. And in fact, there's already somebody doing this. So sometimes I refer patients to this company here, Atomwise, because they're using deep learning to do really powerful docking simulations to find. So if you have an identified target, they will help you find the space of molecules that will hit that target. I think they do it way better than any prior approach to date, but I'm sure you can do way better even still. But now we have to move on to medicinal chemistry, because Prevacid, while it is an inhibitor of this target, it's not a perfect inhibitor. To deliver sort of full inhibition of that target, you would need to be taking thousands of pills a day, and thousands of pills of Prevacid would kill you. It would give you a condition called hypochlorhydria, which you definitely don't want. So what you need to do is modify Prevacid so it's more potent for its NGAs inhibition activity, maybe even knock out the proton pump activity or proton pump inhibition activity at the same time. So now, this is the crazy part, is that we were told that there's no way a drug company would ever take an interest in this disease, and yet Retrophin is doing exactly that. So they said, you've so thoroughly de-risked the science at this point that we will do the medicinal chemistry to actually figure out how to build sort of super Prevacid. But I'm also doing some of that even at Utah with my collaborators there. And so we're taking this PPI scaffold, which we know is the part that really inhibits, and we're generating lots of variants on it to see if any of these will also be more potent inhibitors. And in fact, we already have something that's, I think, about 60 times more potent than Prevacid. So moving in the right direction, but we still have a ways further to go still. And so there's another opportunity here, which is that now that we're generating new molecules, you just have to go all the way back through the approval process. And a key step here is determining toxicity. So I think, human beings have ingested probably billions of different compounds in the course of human history by now. So we should have a reasonably large data set on what human beings can take. We also know lots of things human beings can't take. Maybe we can start to learn in some way in advance which things are likely to be toxic. Because right now, it's basically you blow up a mouse. That's how you do tox. There's got to be better ways of doing preclinical analysis of toxicity, better and faster ways of doing it than the same way we've always done it. So I suspect that the data is there if we're willing to actually learn from it. I think we could do this. And then, of course, there's clinical trials. This is hard, because I thought diagnosis was hard, and finding these compounds was even harder still. But it turns out even more difficult than science is the government in getting this thing actually approved. So, because in some cases we found compounds which should be perfectly safe, but they're not on the market yet. We want to take them, but we can't because they're not through regulatory approval. And it's just very frustrating. So we've been looking for cheat codes here. One of my best cheat codes is actually my wife. So back when we lived in Utah, she went to the state legislature there and got a law approved called Right to Try. And what this does is it gives anybody in the state of Utah who has a life-limiting or life-threatening or fatal condition the ability to take stuff that's been through safety testing even if it's not on the market yet, as long as the company agrees to give it to you. It provides some guarantee that the company won't be punished for doing something like this. Like you can't sue them if they go ahead and give you the drug. But it doesn't protect them from everything. Yeah, so if there's an adverse event, they still have to report it to the FDA, which could jeopardize broader market approval. But it's a step in the right direction towards getting, compounds that people need to be taking to them much faster. And then actually both of us she's testified in the Senate, I have as well. You're trying to get something like this done at a national level. So I've worked in both the House and the Senate trying to shape the laws to say that, the science has moved way past the regulation, and we need better ways of figuring out whether or not somebody should be able to take something. So I think, just to echo what was said earlier this morning, we are needlessly killing people with old-fashioned statistics, and our brutal insistence on doing randomized, placebo-controlled trials. If the data's there, I think we can pull it out and say, look, we don't have to do this to people anymore. And there's honestly a desperate need for good data science when it comes to clinical trials, so that we can come up with better models for these ultra-small patient populations. Because in many cases, the patient population is so small that a priori that even with a pretty large effect, you will not get a statistically significant result, and the FDA will just not approve it, even if it works, which is crazy. So we need better ways. We need techniques that work with lots of data on few patients for pulling out efficacy. So people always ask, how's Bertrand? Well, the good news is he's 9 years old now, and he's doing pretty well. He's a happy kid. It's still a very serious disease. Don't downplay it at all, but he's happy. And I'd say, he's seizure free. He cries tears, and his movement disorder is largely mitigated. He's still certainly facing developmental delay. But as a parent, being able to say that my kid is happy, I think is one of the most important things that I can do for him. So from that, specific execution of this algorithm, I want to sort of briefly highlight the algorithm itself as I envision it. And the goal here is to go from an N of 1, what we did for Bertrand, to do this for more than just Bertrand. And how do we take this to many N's of 1 for all these new diseases that are popping up again and again? And I do see this as an algorithmic process, a process that you go step by step through all the way until you find a treatment. And I won't go all the way through in detail the algorithm. I have a 60-page blog post that I've written on this to encapsulate my thinking so far. If you want that, I can give that to you. But just to show you that there is a process that I've been thinking about here, I'll step you through it, or just in high speed, where you really do move step by step from the patient to their data, and from the data to is this a gain of function? Is it a loss of function? Is it haploinsufficiency? Is it a change of function? Is it amenable to read through? Can you attack the genotype? Do you have to go to the phenotype? It basically, you start branching on what exactly the disease is doing and where it comes from to try to find the right treatment for them. So there's a lot, and I've been pushing other diseases through this model now for the past 2 years. So I'm confident that this actually does work. So yeah, but people always ask, well, can you really do this? And ordinarily, I think it'd be very difficult to answer a question like this, but I just sort of ended up at the right place at the right time, and I think it was the opportunity of a lifetime to figure it out. So this is a few years ago, when he said, President Obama said, he'd seen what I'd done for Bertrand. He said, I'm about to set up this new thing called the Precision Medicine Initiative. I'd like you to be a part. I'd like to see if we can really actually do this, what you've done for your son, and practice for the rest of the country. And I said, sure. He said, will you help me? I said, yeah, obviously I'll help you. Anything you want. I mean, if you want me to go stand on a chair, I'll stand on a chair. Just tell me what you want me to do. The crazy thing is, with these sort of resources behind it, we actually could, really try this out. So I ended up serving as an architect on the Precision Medicine Initiative, which was announced shortly after that. Which is in many ways laying the scientific foundation for precision medicine. By that I mean building the data we need to drive the learnings that will actually enable us to do precision medicine in the clinic. So this program is enrolling 1 million people from across the U.S. and collecting their phenotypic data and their genotypic data so that we can start to figure out, what do these genes actually do? So there's a huge data set being built, whether you know it or not, that will be in desperate need of people who want to learn from it. And in fact, already today, the Million Veterans Program, sort of a precursor to this and a twin to it, has over 500,000 veterans enrolled. So it's already a massive data set available for whoever wants to use it to really become, I think, what I would call the Rosetta Stone of the human genome. I think it'll finally be the